Okay, good afternoon, everybody. My name is Alessia Cuccurullo. I'm here presenting my research, Advances in the Enzymatic Stabilization of Ethanol Construction Material, a research that has been developed during the past 2 years and a half in the Université des Pau et des Pays de la Dour in France and Durham University in England. So, as I said, I'm going to give you a brief introduction just to let you understand the context, and then we will move on with some results. So I'm afraid that it's not a secret that nowadays we are looking for new sustainable and energy efficient construction materials in order to comply with current targets to reduce carbon emissions and energy consumption worldwide. So it's in this context that we talk about the use of rare earth as a building material. And here in this slide I'm just showing some example of the use of earth as a building material. Ethanol construction is not something new, but what is interesting is that these buildings that are aged 100 years, something more, they are still in use and exhibit an excellent performance in terms of mechanical performance, durability, and environmental comfort. So in my research, I started making bricks made of earth. The material object of the study has been provided by the brickwork factory Brissette from the region of Toulouse. If we look at the soil characterization, we can immediately understand that we are dealing with a material that is essentially a silty clay soil. But what is interesting, and that's what I want you to get from this slide, is that our material is suitable, we can say so, for the manufacture of compressed earth bricks according to the current recommendation and guidelines that we can notice here. Another thing that I want you to keep in mind from this slide is the nature of the clay fraction characterizing the soil, because we know that the hygromechanical properties of the material, they are strongly affected by the nature of the clay fraction. And the previous studies about the mineralogical composition of the soil confirmed by my studies show that we are dealing with a kaolinite clay fraction that actually is suitable for ethanol construction because of the low specific surface of this clay that doesn't swell or shrink upon wetting and dry cycle. So in my research, I proposed, according to previous study, a very innovative compaction technique that here is referred as hypercompaction. Because we apply a very high compaction pressure, 100 megapascal, and this pressure is applied statically by using a load-controlled Zwick press, and is applied by 2 aluminum pistons that acts at the bottom and top extremities of the specimen, and it's thanks to this double compaction that we assure that there is no friction between the mold and the soil, and we assure also a uniformity of stresses into the soil. So here on the sides I present the dry density values that are measured on samples that are compacted at different water content. So as the plot shows, as expected, by increasing the compaction pressure from Proctor standard to 100 megapascal, there is a shift of the compaction curve towards higher dry density values. Meanwhile, the optimum water content strongly decreases. So as I said, I'm messing up. Okay. So as I said, I started making bricks of art, and actually the research was quite promising. Just trust me when I tell you that we were obtaining very encouraging results in terms of mechanical performance, hygroscopic properties of the material. But one problem was clear from the very beginning. We needed to improve the durability properties of this material against water erosion. No, I don't want you to believe me, but I'm going to show you. So we performed some durability tests, starting by immersion test. So we prepared small hypercompacted cylindrical sample and we merged them into water for 10 minutes. And then by checking the initial mass of the material and the final mass, we calculated the percentage of mass loss. So now this is our sample after equalization, before immersion. And this is the same sample after equalization, after immersion. So there was a percentage of mass loss equal to 13%. What does this mean? If we try to classify it, we are talking about a material, class 2. Okay, what does this mean? So it's a material that we can use just for external wall, that they are not directly exposed to weathering, or internal wall. Okay, let's perform other tests. Suction test, because we wanted to understand the performance of the material to water absorption. So, and actually we found out that the response was very weak, with cracks and irreversible deformation already after half an hour from the beginning of the test. So still again, a material class 2. Again, a material that we can use just for internal wall. Finally, we performed some drip tests because we wanted also to understand the response of our material to the action, for example, of rain. And so here the results in terms of depth of pitting. And we can conclude that at the end of our test, we made a hole in our brick with a depth of 5 millimeters. What this means? So we have to refer to an index of erodibility that actually is connected to the depth of pitting. And so we can conclude that our material is erosive, slightly erosive. So I hope that I convinced you that our material needed to be improved in terms of durability properties against water erosion. So now the question was easy, and this is where my research came, how we can do that. Let's talk about this. This is more interesting, relaxing. Coral reefs and seashell. Why I'm presenting these two pictures? Because actually, if we look at this, they are magnificent structure, very strong, and they are made up like they are calcium carbonate minerals. And so actually, this was the source of inspiration of a very well-known method of stabilization, well-known like since 100 years, that is called the microbally induced calcite precipitation. In fact, under certain chemical conditions, this particular bacteria called Sporosarcina posteriori is able to induce the precipitation of calcium carbonate. And as the name suggests, it's not really original. What we are trying to do is that we induce the calcite precipitation into the soil to bond the soil particles. This works well when we are dealing with a sand, but I think that now it's clear to you that I'm dealing with a finer soil and an hypercompacted soil. So believe me when I tell you that there is no space for this bacteria into my soil. The size of this bacteria is more or less around 300 nanometers, but anyway, it's too big. So what we can do, actually the responsible of the calcium carbonate precipitation is not the bacteria, but it's the urease enzyme that is a product of the metabolic activity of the bacteria. So actually I thought, instead of using the bacteria, I can use just the enzyme. This is what I need. And the size of the enzyme is just 12 nanometers. So it can fit. And if we can use just the enzyme, we don't have to care about the cultivation and the fixation of the bacteria, all the problem that Emmanuel had, essentially. And so we are talking now about another method of stabilization that essentially the chemistry, the base is the same, but we are using just the enzyme, and the method is called enzymatic induced calcite precipitation. Very original as well. So this is the stabilization reaction. Looks complicated. For the moment, I'm going to explain this later. For the moment, I just want you to focus on the three must ingredients that we need. Urea, the urease enzyme, and a source of calcium ions that can be calcium chloride in this case. Okay, the first question, where to find the enzyme? Because actually this enzyme can be synthesized as, can be found as a synthesized product from chemical suppliers, but is very expensive. But hopefully it is available in a big variety of plants and beans, for example, soybeans, jack beans, pumpkin seeds. And in my research, I wanted to propose a cheap, sustainable way to obtain this enzyme. So I proposed the preparation of a crude urease extract. How I do this? I collect soybeans, and the soybeans are soaked in water for at least 24 hours. And I use 10 milliliters of water for each gram of dry soybeans. Then after the soaked soybeans, with the water they were soaked in, are centrifugated for 3 times into a centrifugation machine, and then I just collect my juice that, in my opinion, contains the urease enzyme. Now, this was never tried before, so I had to prove that in that juice there was the urease enzyme. Okay, so let's try now to understand how the stabilization reaction develops, how it works. So this is my urease extract prepared as I explained in the previous slide, that for me contains the urease enzyme. I had urea. This is the first part of the stabilization reaction. So when I had urea, the hydrolysis of urea starts, and we have the release of ammonium ions and carbonate ions. And then there is also a raise in the pH of the solution. So we have to imagine our solution now full of carbonate ions. This is what matters. Then we had calcium chloride. There is the dissolution of the calcium chloride into our solution. And we have to imagine that now in our solution there is also a big amount of calcium ions going around. Calcium ions, they react with the carbonate ions from the previous reaction, and voila, we have the precipitation of calcium carbonate and a decrease in the pH of the solution. So, looks easy. Of course, there are some parameters affecting the precipitation reaction. So we prepare our urease extract that has some properties in terms of pH, in terms of electrical conductivity. We don't have to forget that there is an enzyme inside there. So we repeated our stabilization reaction by controlling 2 factors, the pH and the electrical conductivity, because actually from the measure of the electrical conductivity we could understand the activity of the enzyme. So we had urea. So what I'm presenting here is actually because we wanted to understand the influence of urea and added to our extract. So we performed like different test tubes by changing the concentration of urea added. So what we got is that for all the concentration that we tested, there is a range in the pH. This is more or less constant for all of them. What is different is actually the electrical conductivity. So it means that it's a kind of intuitive. I mean, like if we had more urea, maybe we have more activity of the enzyme. This is what maybe we were expecting. So we left this test tube for 24 hours. And here we can start to see a difference. So the pH stay constant, but what's changed actually is the electrical conductivity. Because we have to imagine that with time that is passing, more urea is being hydrolyzed. And then we had calcium chloride. And what we can see it's a decrease in the pH of the solution. We don't report here the measurement for the electrical conductivity because what we were measuring the electrical conductivity just to detect the activity of the urease enzyme, so it was not worth at this point. This is how, these are our test tubes, how they look like. So how we can notice is that what I report here is like that the amount of precipitated calcium carbonate is not the same for each like test tubes. And so what we did is that we filtered each test tube, we collected the material, and we oven dried it at 40 degrees, not 105, because maybe there was some organic matter present in our precipitated material. And this is what our precipitated material look like. It really looks like calcite, but we cannot say it from the very beginning. So we performed an XRD test to have a proof that the precipitated material was calcite, and here's our proof. So this actually the result of this test proved us that what we obtained was calcium carbonate in the form of calcite and vaterite. Okay, and then after we also measured the amount of precipitated calcium carbonate. And in this plot, we showed the theoretical amount of precipitated calcium carbonate and actual amount of precipitated calcium carbonate. There is a difference that we can notice. So what we can say is that it is not true that if we had more urea or more calcium chloride to our solution, we obtain more calcium carbonate in the end. Because actually there is an influence of the amount of urea added to our solution on the activity of the enzyme. And there is a kind of inhibition of the enzyme if we had, after a certain point, if we had more urea. And so this is what actually we wanted to find, our optimum, like how much urea I have to add to my solution to obtain like the highest amount of precipitated calcium carbonate. So I want just you to look at these three graphs. So here, like the visual examination of our test tubes. Here, the calculated amount of calcium carbonate, and on the top, on the right, it's the urease activity, by measuring the electrical conductivity. So they have more or less the same shape. So the amount of calcium carbonate is strongly related to the activity of the urease enzyme. The application to the soil was not easy. We tried many things because we didn't know how much water to introduce in our soil. It was not enough because, of course, to obtain more calcium carbonate, you needed to add more water, but then after this was not compatible with the compaction procedure. So that was a little bit tricky, but in the end, I can show you some of the results that we achieved. So here is a reference sample. It's a standard Proctor compacted sample. So before immersion in water. This is the same sample, not stabilized after immersion. And there is a percentage of mass loss equal to 42%. Okay, now the question is, if we apply our method of stabilization to my sample, what happens? We try to stabilize it, we perform the same test, and this is what we got. So in the end, we reduced the percentage of mass lost to 13%. And then we also performed some XRD analysis to have a confirmation of what that was because of the precipitation of calcium carbonate into our soil. And so this is the XRD for the no-stabilized soil. There is calcite in this, already in the soil. And this is the XRD for the stabilized soil. It doesn't really make sense if you look at the graph like this. I just want you to keep in mind one thing. So, yeah, if we look at the calcite peaks, all of them, there is like a trend that is repetitive. And for example, for the unstabilized and the like soil, it's like the black line. I don't know if you are able to see it. You are able? And instead, for the extract stabilized soil, we can notice that the peak is like higher. And this is for all the peaks. Instead, if we have a look at the kaolinite peaks, we have like the contrary behavior. So there is a decrease. And my guess is that so in this soil, we are having like, there is like a precipitation of calcium carbonate, and there is also another thing that we are doing is alkaline activation. So here, this is why we have the dissolution of the clay minerals. In conclusion, I just want to say that I think that I proved you that there was a need to improve the durability properties against water erosion. We proposed a method of stabilization. Preliminary test, it's a method that it's not really, it's still at the beginning, how to say, and there are so many tests that need to be run. But of course, I mean, I think that I give you a proof that we can work on this method to improve the durability properties against water erosion of our soil, and so make like earth still a solution as a building material. And here a list publication if you are interested to have a look at this method. And thank you for the attention. Question if